Hi, I'm Maggie, and I'm a student at Jefferson Institute for Bioprocessing, and I'm currently enrolled into the master program. Coming from a biotechnology background degree in undergrad, I really wanted to see how I would be able to implement the knowledge I learned in undergrad into the industry. And when I found out about this program, it really brought together a lot of the basic understanding I had of biology and biologics, and I was able to put that into industry perspective. And professors here are great, and in particular, Parvis, the director of this program, is very enthusiastic about his work and the bioprocessing field, which really motivates us as students to dive into the topic and learn more about it and be really interested into each of the topics that he's taught us. Currently, the gene and cell therapy space is the next up-and-coming next generation therapeutic, and so we are trying to look more into the space for influenza recombinant vaccines, where we will do DOE studies for the upstream processing and downstream processing of influenza vaccines.
So this is the active pure purification system, where it allows you to separate your protein of interest. In our case, this is an antibody from your other impurities that you may have in your solution. This is the cation column. It has negatively charged particles. In the column you see here, it has an attraction towards positively charged particles, which in our case, the protein of interest is an antibody. After the antibody has attached to the negatively charged beads, we can then elute our protein of interest using a salt gradient.
And being in this program really gives me a big picture of what the biopharmaceutical industry is like, because you really dive into each of the unit operations and learn the details and the concepts behind it, and we are able to bring that concept into the lab and actually perform several experiments on the topics that we discussed in class.